There is nothing to be disclosed. What is cardioneuroablation? Let's see some basic concepts. We created the cardioneuroablation in the 90s and gave it this name to make the technique easier to be understood. The aim was to get vagal denervation by endocardial radiofrequency ablation. Here we have a simple schematic view of the cardiac autonomic nervous system consisting of pre- and postganglionic neurons. The parasympathetic system is millions of years older than the sympathetic one. Only the parasympathetic neural body is located in the atrial wall, and only this can be eliminated by endocardial radiofrequency energy. In normal condition, this neuron releases acetylcholine that acts on muscarinic receptors in the atrium, causing extreme cardioinhibition, transient cardiac arrest, and a huge atrial refractory dispersion. It is responsible for the cardioinhibition that causes syncope. The muscarinic effects are completely eliminated by atropine. Sympathetic and sensory neurons are away from the heart and protected from radiofrequency. Therefore, they are preserved, and their fibers recover in a few weeks. After cardioneuroablation, there is an important and persistent chronic reduction in both the parasympathetic and the sympathetic tone. In the 90s, by using spectral analysis online, we found that the normal heart has two kinds of myocardium: the compact, that is composed of electrically very well attached cells, and the fibrillar one, that is composed by poorly connected cells. Clusters of fibrillar myocardium we named as atrial fibrillation nests because they are directly related to the atrial fibrillation substratum. The atrial fibrillation nest may be easily mapped with spectral mapping or even by conventional recordings with specific filtering. Despite having large spectral difference, electrical potentials of fibrillar and compact myocardium may be relatively similar in sinus rhythm. However, after atrial fibrillation induction, there is a great difference, with fibrillar myocardium showing very high frequency, disorganized and irregular electrical activity, whereas the compact myocardium shows a lower frequency, regular and very organized electrical activity. Assuming that the atrial fibrillation nest would be directly related to the atrial fibrillation substratum, we decided to ablate the atrial fibrillation nest, trying to treat the atrial fibrillation. There were two interesting results. First, the atrial fibrillation nest ablation made the atrial walls highly stable, and after this it was very difficult to reinduce the atrial fibrillation by using high-frequency atrial pacing. The second consequence of atrial fibrillation nest ablation was intriguing. We observed that after this ablation, there was a very important and persistent vagal denervation. In this case, for example, the heart rate variability was drastically reduced from 163 milliseconds of SDNN before ablation, and it went down to 78 milliseconds even 2 years after ablation. Considering that the ablation of the atrial fibrillation nest caused the vagal denervation, we realized that atrial fibrillation nests are related to the cardiac innervation entry, and that the presence of interlaced neural fibers causes a certain degree of electrical disconnection between myocardial fibers, causing changes in the spectrum. Also, the electrical poor connection allows the electrical resonance that helps the atrial fibrillation maintenance. Here we see an electroanatomical model tagging atrial fibrillation nest using the fractionation mapping. We developed this software in 2005 to aid the mapping of the innervation. In this case, the gray areas present high probability of innervation and should be ablated, while purple areas have low innervation probability and must be not ablated. During cardioneuroablation, longer radiofrequency applications are required in the presumed areas of the 4 main ganglionated plexi. The place 1 between the superior vena cava and the aorta, the place 2 near the insertion of the right pulmonary veins, the place 3 in the region of the coronary sinus ostium, and the place 4 near the insertion of the left pulmonary veins. Is the cardioneuroablation able to reach the epicardial GPs? Yes, it does. The RF lesion can reach 15 millimeters in depth, and there are reports in the literature of terminal injury of the esophagus. Thus, there is no doubt that well-targeted applications may eliminate epicardial ganglionated plexi. Here we have a map reaching complete denervation and elimination of the vagal response. The most important denervation point is called P point, because it gives origin to the most of the innervation of the P cells and of the sinus node. It is located in the left interatrial septum between the insertion of the right pulmonary veins, roof of the left atrium, and fossa ovalis. The GP2 area is usually near, located at the insertion of the right superior pulmonary vein. The GP3 area is typically assessed at the insertion of the coronary sinus, as well as the region of the Marshall's vein in the roof of the coronary sinus. The GP4 area is close to the insertion of the left pulmonary veins, and finally, the GP1 area is assessed by the superior vena cava. In some cases, may be necessary ablation of the Waterstone groove and the other areas indicated by fractionation mapping until vagal effect elimination. How to study the vagal innervation during atrial fibrillation ablation? To study directly the vagal innervation, we developed the extracardiac vagal stimulation. In this method, we advance a catheter through the superior vena cava and internal jugular vein up to the jugular foramen. This point is too close of the vagus nerve, allowing non-contact vagal stimulation that causes asystole or high-degree AV block. This maneuver can be repeated as many times as necessary during electrophysiological procedures. In the lower tracing, we see that there is an intensive vagal effect, but after the ablation of the atrial fibrillation nest, the extracardiac vagal stimulation was repeated and there is no longer any vagal response. In this study, my brother and I sought to verify the relationship between atrial fibrillation nest and vagal innervation. We compared 2 groups of patients undergoing atrial wall ablation. In the first, we performed the ablation of the atrial fibrillation nest, and in the second, the ablation was outside of atrial fibrillation nest, targeting compact myocardium only. Through the extracardiac vagal stimulation, it was verified that after atrial fibrillation nest ablation, there was complete elimination of the vagal response, whereas in the group submitted to ablation outside of the atrial fibrillation nest, the vagal response was completely preserved. These findings reinforce the idea that the atrial fibrillation nest are related to the entry of the vagal innervation and may be used as a kind of innervation mapping. Thus, considering that it was possible to map the atrial fibrillation nest and to get vagal denervation, we proposed the cardioneuroablation with the aim of treating functional bradyarrhythmias and even the atrial fibrillation. Let's now see the role of cardioneuroablation in the atrial fibrillation ablation. In this example, we show the relationship between vagal innervation and atrial fibrillation. We see an extracardiac vagal stimulation that causes sinus arrest, followed by the spontaneous onset of atrial fibrillation. After elimination of the acetylcholine, the atrial fibrillation reverts to sinus rhythm. This example shows how vagal innervation is highly arrhythmogenic to the atrial walls. This is probably while the nature removed most of the vagal innervation from the ventricles. After cardioneuroablation, the extracardiac vagal stimulation is repeated. We can see that there is no more induction of bradyarrhythmia and there is no more induction of the atrial fibrillation. It's possible to see also that the heart rate was increased because the effect of the cardioneuroablation is an elimination of the vagal tone that causes obviously an increase of the sinus rate. Now let's see the determination of the atrial refractory period during vagal stimulation. In the upper tracing, there is the determination of the atrial refractory period without induction of arrhythmia. After this, we again repeat the determination of the atrial refractory period during vagal stimulation. In this case, there is immediate induction of the atrial fibrillation, and we also observed an important reduction of the atrial refractory period caused by the vagal effect. After the cardioneuroablation, we again repeat the determination of the atrial refractory period, and there is no more induction of the atrial fibrillation. So, the disappearance of the atrial fibrillation induction during the termination of the atrial refractory period during vagal stimulation is nowadays our endpoint to get the best result for atrial fibrillation ablation. The extracardiac vagal stimulation has showed that the vagus causes a huge reduction of the atrial refractory period. So, let us now analyze the intense tissue effect of the vagus causing dispersion of atrial refractoriness. Let's consider two similar myocardial cells, A and B, that have an atrial refractory period of 208 milliseconds. Let's now add the vagal effect. Due to the lack of the motor end plate, the A cell will have strong acetylcholine action, while the B cell will have no cholinergic effect. In this way, the action potential of cell A is reduced to 8 milliseconds, and that of the cell B is maintained, resulting in a huge refractoriness dispersion of 200 milliseconds. This phenomenon is one of the main causes of the atrial fibrillation bursts and can be corrected by cardioneuroablation. Let's see now the effect of neurotransmitters on the induction of atrial fibrillation. In this elegant study, Sharifov et al. Injected adrenaline into the sinus node artery, and the atrial fibrillation was induced. Apparently, adrenaline causes atrial fibrillation. However, if adrenaline is injected after atropine function, eliminating the vagal effect, atrial fibrillation is no longer inducible. This demonstrates that adrenaline produces atrial fibrillation when there is a vagal effect, but if it is lone, it does not cause atrial fibrillation. On the other hand, when acetylcholine is injected into the sinus node artery, there is always an induction of atrial fibrillation, demonstrating that acetylcholine is highly arrhythmogenic. Does the cardioneuroablation denervation last? Yes, it does. In a study of 83 patients undergoing cardioneuroablation and followed by 2 years with Holter, we found an important reduction in the parasympathetic and sympathetic innervation by using the heart rate variability in time and frequency domain. There was a significant increase in the minimum and median heart rate, but there was no change in the maximum heart rate. There was a significant reduction of 34% in the SDNN and 85% in the PNN50. There was also a reduction in all parameters in the frequency domain from 71 to 8%. This data show that cardioneuroablation promotes a long-term persistent reduction in both the parasympathetic and sympathetic innervation. So, based on these findings, we have been observing positive effects of cardioneuroablation in the atrial fibrillation ablation setting. The first one, the cardioneuroablation abolishes the vagal atrial fibrillation reinduction by vagal stimulation. Second, the cardioneuroablation prevents the vagal shortening of the atrial refractory period. Third, the cardioneuroablation reduces the atrial refractory dispersion. Fourth, cardioneuroablation prevents the atrial fibrillation induction by extra stimulus during vagal stimulation. And fifth, cardioneuroablation chronically reduces the sympathetic and parasympathetic tone. The gold standard for atrial fibrillation ablation is the pulmonary vein isolation. However, to verify the value of cardioneuroablation in atrial fibrillation ablation, we carried out the following study. 127 patients with paroxysmal or persistent atrial fibrillation were included, shared in two groups. The first with 55 cases submitted to classical pulmonary vein isolation, and the second one with 72 cases submitted to pulmonary vein isolation plus cardioneuroablation, being the endpoint the elimination of the vagal response confirmed by extra cardiac vagal stimulation. In the mean follow-up of 22 months, we observed that the group in which we performed the denervation, there was 5.38 times less recurrence of the atrial fibrillation in the long term. The hazard ratio was 5.38 with a highly significant statistical P of 0.003. This data suggests that cardioneuroablation can significantly improve the pulmonary vein isolation results. And also, we can observe that probably the denervation in treatment of the atrial fibrillation ablation is very important. In conclusion, we may say that the vagal innervation is highly related to the atrial fibrillation origin, being clearly arrhythmogenic. The addition of cardioneuroablation to pulmonary vein isolation, confirming the vagal denervation by extracardiac vagal stimulation, significantly increases the success rate of the pulmonary vein isolation with a remarkable reduction of long-term atrial fibrillation relapses without increasing complications. Consequently, in atrial fibrillation ablation, the ablation of the veno-atrial junction as well as the neuroatrial junction seems to be essential. Thank you very much. I wish all of you a wonderful Congress.